Stay on for the next talk because the next speaker is Jillian McKee from CHOP, who's going to be talking about looking at qEEG for development of a biomarker for STXBP1.
All right, thanks Jim for the introduction and for everyone for having me here to speak today. As Jim mentioned, I'm Jill McKee. I'm a pediatric epileptologist at CHOP, and today I'm going to be talking about some of our work trying to identify quantitative EEG biomarkers of STXBP1. All right, so there's three main points I want to get across today. And the first is, can we find an objective EEG-based biomarker to help distinguish individuals with STX from neurotypical controls? The second is, can we assess whether these EEG features correlate with the developmental outcomes that we actually care about? And then the third is, how stable are these biomarkers over time? Are they consistent? And then can we use them to actually take it one step forward and predict outcomes in patients? All right, so I don't want to belabor the point because lots of people have been talking about biomarkers before me, but I like to think of them as having these three main components, which I kind of already mentioned, but biomarkers can be diagnostic, meaning they can be like a unique signature of a disorder and help distinguish people with that condition from control patients. They can be prognostic or predictive, meaning they can track with the severity and can be used to stratify patients. And then finally, and most importantly, they could be responsive. And this is really what we want for a clinical trial, right? We want a biomarker that we have a reasonable basis to believe that it would actually respond with treatment. So I don't want to spend too much time going through background. Most people in this room are probably already familiar with what EEG is, but it's just a way to measure the electrical activity in the brain. And it's a clinical diagnostic test, and it's used both in clinical care for patients and then also in research. And when we use EEGs in clinical care, we tend to visually interpret them, right? We plot the voltages on little charts like you can see here, and the epileptologist looks at it, and it can look for the background organization, look for things like epileptiform discharges, look for seizures, and then look for slowing either globally or in specific regions of the brain. And that can tell you something about how your patient's doing clinically. But we can learn so much more from EEG tracings when we take a computational approach. So in addition to those clinical signatures I previously mentioned, we can look at spectral features. We can look at spatial or topographic measures. We can look at event-related potentials, such as visual and auditory evoked potentials. We can look at different sleep or state-based measures, functional connectivity, and then more complicated things like temporal and nonlinear dynamics. And what I'm going to be focusing on today is spectral features. And what that means is you take the EEG signal and you can decompose it down into its underlying frequency components. And in EEG land, we call these different frequency components delta, theta, alpha, and beta, going from slower frequencies to faster frequencies. And any signal you can take it apart and look at how much is made up of delta versus theta, and that's called the power in those frequency bands. And then we can look at the ratios as well. So when we commonly look at, and I'm going to show you later, is like alpha theta ratio, or you can look at delta theta, or any ratio between one power in one frequency band to another one. And we can do this both globally, looking across the whole brain, and then regionally, looking at specific areas of the brain. All right, and then one final last piece of background I just want to share is for these results I'm going to show you here, we're using two different types of EEGs. We're both repurposing clinical EEGs, so these are EEGs that were obtained in our patients with STXBP1 and neurotypical controls for clinical care, and we're reusing them to look for spectral features. And then we also have prospectively collected research EEGs that we're getting as part of the STAR Natural History Study, which you'll hear more about later. But there's a couple differences between these EEGs. They're completely different recording setups, different numbers of electrodes, different frequency sampling of the signals. So just a couple caveats to keep in mind. All right, so let's dig in now to the data and start. I'm going to show you how we can develop EEG biomarkers that are diagnostic of STXBP1. So we did this first using our clinical EEGs, so just repurposing clinical EEGs, and we built a EEG extraction pipeline and processing pipeline to get the data clean, and then pull out the power in these different frequency bands. And then what we did was we trained machine learning models to distinguish whether specific EEGs came from a neurotypical control or came from an individual with STXBP1. And you can see the ROC curve in the middle of this plot, this figure here, and it performs pretty well. But that's not really what we care about. What we care about is having a biomarker of STX, right? We already know whether these kids have STXBP1 or not. But the nice thing about this approach is we use random forest models, and then we can look at the feature importance and pull out which features did the model use to make that decision. What was driving this categorization decision? And what we see for STXBP1 is that the strongest predictor by far is the alpha-theta ratio in the frontal region of the brain. So that suggests that this could be a good potential biomarker for STX. And then when we look further, what I'm showing you here is neurotypical controls in white, clinical STX EEGs in the middle blue color, and then in the lighter, brighter turquoise color, sorry, I should have picked different colors, those are hard to name, you see the research EEGs. And then what you can see is after about age 3 or so of life, we see consistent decreases in frontal alpha theta in individuals with STX compared to neurotypical controls. And the really nice thing about this is it's consistent across these two completely different recording systems, different patients, different populations. All right, so now we have a potential biomarker of STX, but how does that relate to outcomes? So, right, we want this biomarker to be prognostic or predictive and be used to stratify patients and track with severity. So the nice thing is because we are running this large natural history study where we are collecting quantitative EEGs every 6 months, we're also getting a whole battery of developmental assessments at the same time on the same patients. So that gives us a nice opportunity to look and see how these EEG features correlate with outcomes. Oh, and just for anyone who wants, you're going to hear more about the STAR study later, but for anyone who wants to know about more about our protocol, just want to put a plug that our protocol papers just came out published online just a couple days ago, so please check it out. And then here's some of the developmental trajectories that we're gathering data on in STAR. So here you can see the 5 subdomains of the Bayley, the CARS, or Childhood Autism Rating Scale, the GMFM, which is a gross motor function measure, the 2 fine motor domains of the Peabody, and then some of the SCSA scales, which you're going to hear more from Megan about later today. But I don't want you to focus too much on this data, just to show you that we have a lot of this data time-locked to EEGs, and we're going to look at how it correlates. So here is a very big heat map showing all of the EEG spectral features along the x-axis, and then the developmental measures along the y-axis. And what we're showing you here is just the Spearman correlations from the baseline assessment between all of these EEG features and the developmental outcomes. And this is work that's largely being done by Ian McSally, who's sitting right over there. And what you see is here I'm pulling out our frontal alpha theta, and you can see that it has a lot of strong correlations with developmental measures. The 2 strongest being the 2 motor domains of the Bayley, the fine motor and gross motor. And when we pull out here the Bayley fine motor, here I'm going to just show you a blow-up of the correlation. So those prior correlations were just taking the baseline assessments. And one other thing we've done is try to factor in all of the longitudinal data. And we see that these associations hold when we model this longitudinal data and longitudinal EEGs using mixed effects modeling. And just want to put another plug in for the SCSA. This was interesting to us, but using this modeling approach, the SCSA is actually very, very highly correlated with several of the quantitative EEG spectral features. All right, and then what about stability and outcome prediction? So we want a biomarker that would be responsive to treatment, but we obviously don't have any disease-modifying treatments that are currently available now to assess whether this is the case. But there are a couple of prerequisites we want to make sure our biomarker follows. For biomarkers that you want to use in a trial, you really want them to be stable and you want them to be reproducible. So saying it another way, when the patient is stable and not changing, you don't want the biomarker to change. And then when the patient does change, you want to see a change in the biomarker. And you ideally want something that looks kind of like this green plot here, where it's stable at baseline, and then after you give a treatment, you see a change. And this matters for clinical trials for several reasons, right? It gives you more confidence that any change you see is real. It gives you better sensitivity to detect treatment effects. It can improve the interpretability of your biomarker. And then if you're designing a trial, it can help you potentially design smaller, more efficient trials. So this is something we've just started looking into now because we actually have now 3 years of longitudinal data. So we have, this is a little old data pool, but we actually have now up to 6 EEGs per participant. And we can look and see how stable are our EEG quantitative features. So here I'm showing you again that frontal alpha theta for each participant over time aligned to their first assessment. So people come back typically every 6 months, so you can see individuals are connected with a line. And what we see is that it has pretty good intra-individual stability, where we see an intra-class correlation coefficient of 0.76, which is actually pretty good. But then if you compare that to the GMFM, which is very, very stable, you see that, I'm showing you the same plot here for the GMFM, we have a correlation coefficient of 0.93, so it's very stable over time. All right, and then the last thing we wanted to look at was, can these EEG biomarkers actually be used to predict developmental outcomes? So we've now learned the relationship, we've looked to see how well they correlate. Can we make predictions about what a patient's developmental level should be based on their EEG features? And we did this 2 ways, both across the whole population. So like, imagine you have like a new trial participant. Can you, based on their EEG, predict what their developmental level would be? And then we also did it at the individual level, trying to predict what an individual participant's trajectory would be. And that's what I'm showing you here. So on the left, you can just see the correlation between the GMFM66 and the frontal alpha theta EEG biomarker. And then in the middle, you can see the predictions of the GMFM based on comparing the predicted GMFM to the observed GMFM based on using the alpha theta in the frontal region. And you can see that it's not that strong a correlation there. But when we look within participants, we actually get a very strong correlation with a Spearman's correlation coefficient of 0.92, which it's promising for future trials that there is some stability of these features and that we're able to predict developmental outcomes using them. All right, so in summary, hope I've been able to convince you of 3 things: that we have some useful EEG-based biomarkers of STXBP1, that these biomarkers actually correlate with developmental outcomes, and then finally that they're stable and have potential utility for predicting patient outcomes. All right, so I want to thank our huge team, especially Ian McSally, who's done a lot of the EEG analysis, and then Peter Gaylor, who originally developed our quantitative EEG pipeline. And then want to thank all of our funders, and then of course thank the families for participating in our studies.
Dr. McKee, outstanding talk and very interesting work. Could you go back 1 or 2 slides to the population and the subject specific?
Yeah.
I wonder if you can go into a little more detail about those last 2, how the underlying stats are different. What did you do differently? The correlation is so compelling on the right, but I don't quite get how you get from the middle to the right.
Yeah, so it's using the subject specific is using data from that individual participant to try to predict their next one. So you have, the model knows their prior GMFM as well. So it's a little informed, it knows like their baseline, where they're starting from, right? Because if you, here, like, where's my, so like if you can see here from like the GMFM, they're all aligned and they're pretty stable, but they all have a different y-intercept. You're essentially, the model knows the y-intercept in the second one.
Got it.
Yeah, out of curiosity, so this, I guess the alpha theta in the frontal regions, that's satisfyingly specific. And so I sort of wonder, like, you know, one implication might be that, like, I really want to make sure I get great pharmacodynamics and a great effect in the frontal region in order for that to be affected or to see rescue. Do you actually think that is true? Like, does that pattern actually depend more locally, or even if I had poor biodistribution or a lot of effect elsewhere, that would actually affect globally the brain so that you would see that kind of change in the biomarker?
Yeah, the short answer is we don't really know. I think it is still would be prudent to have like a broad biodistribution cortically, but that's a potential thing that maybe you do want to make sure you have frontal. I mean, the frontal lobes are very important for cognition and motor and everything, so I think you would definitely want to have biodistribution there.
But I guess, do you think the signature is actually affected by, I mean, it is a cumulative effect of like the entire brain's circuitry, or do you actually think it's very important, like that pattern is generated locally and therefore it might depend on a more local
I think we don't really know.
Okay,
Yeah.
thanks.
Great talk.
Thanks.
I guess kind of two comments, similarly related. Narrowed in on the frontal alpha theta because that was discriminatory in comparison to controls, but that may not actually be the same thing that is discriminatory within severity.
That's definitely true. Yeah, and even if you look at even our outcome measure correlations, there were a lot of things that correlated, right? Oh, it's going to take me forever to get back to it. But like here or here, it's a little hard to see, but all of those asterisks were significant correlations, and some of them with other spectral features are actually stronger than with the alpha theta.
Yeah. So I was wondering about kind of using the machine learning, but to discriminate based on
Between levels?
Between levels
Oh, yeah.
rather than that, to sort of see if that comes out with a similar conclusion.
Yeah.
And then
No, that's a great suggestion, and now we actually probably have almost not super fine-grained levels, but broad categories. We probably have enough data to begin looking at that.
yeah, and then the other part was it doesn't work under 3 years of age, which actually makes a lot of sense because we have much less alpha
Yeah.
in general developmentally.
Yeah, the main difference is if you look at the plot, like that the controls are getting more alpha as they age and the STXers are not. So yeah, it makes sense that you see it later.
So do you have enough patients in the younger cohorts to be able to start to look at whether there's other signatures that are going to work better in the younger cohort? And
Yeah,
similarly
we
modeling
could.
probably
We
for the
haven't done
severity
that. We do
component.
have fewer people who are
Yeah.
like that under 2, but we may have enough. We could look. We haven't looked yet.
Cool.
Yeah, thanks.
Great work.
Thanks for a great talk. I'm curious, are the measures that are most related to developmental outcome also the ones that are most related to epileptic symptom burden or frequency? Do you have those data?
So we looked at that, and there's not like a ton of correlation with the spectral features and actual epilepsy, either having epilepsy or the presence or absence of seizures or the severity of seizures, which we actually kind of think is a good thing that the EEGs aren't being super contaminated by seizures, because we really wanted a developmental biomarker more than seizures, because seizures are relatively easy to count and measure already, so we didn't really need another way to track them.
Is this specific for STXBP1 DEE, or are you seeing the same in other DEEs?
Yeah, great question.
And what about Delta?
Yeah, so we've looked in several different DEEs, and the frontal alpha theta seems to be more specific for STXBP1. The other condition I primarily work on is SYNGAP, and we see occipital alpha theta reductions in kids with SYNGAP. So same spectral ratio, just different localization, which is interesting. And then delta, we see increased delta in pretty much all of these DEEs. But yes, we have looked at that too.